Now, most of what we're going to be talking about the rest of this lab is how things move around in solution and also how they cross membranes. So we're talking about stuff that is dissolved in a liquid. We're talking about solutions. And we're going to be looking at the particles in solution and how they move around. And everything that we're going to be talking about is going to be based on something called Brownian motion. Brownian motion is the underlying principle for diffusion. Now, there was this guy named Robert Brown. He studied plants mostly, and he was looking at some pollen under his microscope. And to do that, what you would do is you would take some pollen, put it in some water, and then look at that under a microscope. And what he started noticing was even when that specimen that he was looking at, when it was held completely still, those pollen grains were moving around. They seemed like they were vibrating. They were moving around in like this zigzag motion. And there wasn't any rhyme or reason to it. It was very random. And he couldn't figure out what it was at first. And he noticed the more and more and more times that he looked at pollen grains and eventually at other stuff too, it all did the same thing. When we look at particles in solution, which really just means things dissolved in a liquid, when we look at those, they move around. And that became known as Brownian motion because Robert Brown discovered it. Now, most of what we're going to be talking about when we're talking about particles in solution are way too small to be seen. So we may be talking about glucose, or we may be talking about sodium or potassium or something like that. Now, these are way too small to be seen. But pollen, we can see that, and it behaves pretty much the same way. So now we're going to start talking about how particles move around in solution. And when we talk about things moving around, sometimes they do it completely on their own, or sometimes they require energy. When they do it completely on their own without any energy being needed, that's classified as passive transport. And really, that's most of what we're going to be talking about in this lab. In lecture, you'll talk more about the other type, the type that requires energy. And when something requires energy to move, that's called active transport. So passive does not require energy. Active does require energy. And like I said, most of what we're going to be talking about in lab is passive transport, but there's a bunch of different types. The first type, the most basic type, is called simple diffusion. Simple diffusion does not require energy. It is passive transport. And it is simply the tendency for things to move from an area of high concentration to an area of low concentration. Things do that. Now, this isn't really simple diffusion, but there's one way to visualize this. Say you were in a gym and there were about 100 other people, and for whatever reason you were all pushed into one corner of that gym. You're all crowded together, and time goes. Is everyone going to stay crowded together there? No, they are going to spread out so that they are pretty much evenly distributed through that gym. Well, that's kind of how simple diffusion works. And I'm going to give you something that you can actually do at home so you really do get some hands-on experience, but I'll give you that in just a few minutes. In simple diffusion, if we take something and put it down in a liquid, if it's something like, let's say, food coloring, or maybe something that's more common, more easy for you to think about, is a tea bag. If you put food coloring or a tea bag down in a glass of water and let it sit and watch it, enough time goes by, you will start to see either that food coloring start to disperse, or you'll see the oils from that tea start to disperse. So it's going from an area where it's in really high concentration towards an area where it's in really low concentration. When you first drop some food coloring down in a glass of water, that drop that falls in has a high concentration of food coloring, whereas the water has no concentration, so it's a very low concentration of food coloring. And given enough time, that food coloring is going to spread out. If you were to come back in an hour or two and look at that glass, it's all going to be the same color because everything has spread out. And the reason that happens is because of Brownian motion. So if we were to look at one particle of the dye in that food coloring, it's zigzagging around. And the other particle is zigzagging around, and they're all zigzagging around. But when they're in high concentration, when they're all packed tightly together, they're not just zigzagging around, they're bumping into their neighbors. And when they bump into their neighbors, they ricochet, they shoot off in opposite directions. And they keep bumping into their neighbors, and they keep shooting off in opposite directions until they are spread out so much that they're not really bumping into their neighbors hardly at all anymore. So, the closer packed they are, the higher concentration they are, the more they're bumping into their neighbors, until eventually they're so spread out, they very rarely bump into their neighbors. And that's the underlying foundation for simple diffusion. Now, simple diffusion, it occurs without the use of energy. But there are some things that actually can make it happen faster or slower, and that's what's down here. The higher the temperature, the faster the rate of diffusion. When we heat things up, that zigzag motion actually speeds up. And if it speeds up, then they're much more likely to bump off of their neighbors. That's why if you take a tea bag and put it down in hot water versus another tea bag that you put down in cold water, the hot water is going to have that tea spread out through it a lot faster. Cold water, that tea does not move out very fast at all. The next two kind of go hand in hand. The smaller the size of the dissolved particle, and the smaller the weight of that dissolved particle. So really, the smaller something is, the faster the diffusion. And that's just because smaller things can move faster than bigger things. So smaller size and smaller weight both increase the rate of diffusion. And the fourth thing is the more concentrated the particles, the faster the rate of diffusion. And that goes back to what I was just saying about when something is in higher concentration, the more likely it is to bump off of a neighbor. So the more concentrated, the more bumping, the more ricocheting, and the more it will spread out quickly. So if we were in the lab, I would do a demonstration of this. But this is one of the things that you can actually do very easy at home. And I know it's going to sound very, very elementary school, very childish, but this is something that you really can do for lab at home, and I suggest that you do it. I really recommend you do it just so you can say, hey, I did something for lab, hands-on. And that's to take a glass of water. And if you have food coloring, then take a couple drops of that food coloring and drop it into that water. If you don't have food coloring, find a tea bag and drop it down into that water. Don't move it around a lot, just let it sit down in the water. And then now, over the next, half hour to an hour, look at it and watch to see what happens. And you can actually fill out everything here. That's simple diffusion in action. Next is facilitated diffusion. So now we're not just talking about things moving around, we're going to start to actually apply this to our cells. Now our cells are basically like little tiny little balloons, and inside those balloons there's a lot going on, and the actual edge of that balloon would be the cell membrane. Cell membrane is just the outside border of a cell. Now the cell membrane, sometimes called the plasma membrane, lets things in and out of that cell, but it doesn't let everything in and out. That would be bad. There are some things we don't want to get into our cell, or some things that we don't want to get out of our cell. So the plasma membrane is very selective with what can get in or out. So we say that it is selectively permeable. That just means it can let some things cross, but not everything. Sometimes you will also see it called a semi-permeable membrane. Same thing. Now, when something's crossing one of these semi-permeable or selectively permeable membranes, most of the time it's doing that through special proteins on the surface of that membrane. For example, sodium is constantly moving into every cell in our body, but it's doing that through a protein called a sodium channel. This is basically like a tube that goes completely through the membrane of one of our cells, but it's shaped in such a way that sodium is the only thing that can get through. Now we also have potassium, but potassium is constantly coming out of every cell in our body. And it does that through a potassium channel. And potassium channels are shaped so that potassium and nothing else can get through. Potassium won't fit through a sodium channel, and sodium won't fit through a potassium channel. Anytime something needs in or out of our cells, chances are there's going to be a protein shaped to let that substance in or out. So when something is moving in or out of our cells, chances are it's crossing through one of those proteins, and what we do is we call that facilitated diffusion. Now, there are some times when things are moving in and out of our cells, but it requires energy. That is not facilitated diffusion. That is something else, and that's something that's covered in lecture. But when sodium, for example, is coming into our cells, it's because there's a high concentration of sodium outside the cell and a low concentration of sodium inside the cell. So remember, everything likes to go from high to low concentration. So it's still diffusion. It's moving from high to low, but it's doing this across a selectively permeable membrane. So it's diffusion, but that diffusion is requiring some sort of carrier protein. So we call it facilitated diffusion. Next, osmosis. Osmosis is another type of diffusion, but it's got some very, very specific details that set it apart from just plain simple diffusion. Osmosis is the diffusion of water, and it is the diffusion of water across a selectively permeable membrane. So I'm going to actually shrink all these windows down and use a whiteboard to explain this. It's the diffusion of water across a selectively permeable membrane. So let's say I have an aquarium, and that aquarium is divided in half by a selectively permeable membrane. On one side, I've got a sodium solution. So when I say sodium solution, that means that it is sodium dissolved in something, probably water. Now, I don't know if you've talked about solutions in lecture yet, so I'm going to tell you a little bit more about solutions maybe than you need. But solutions have two parts. First, they have something called the solvent. The solvent is usually water. Not always, but usually. Now, the solvent is the bigger portion. There's more solvent than the other half. Now, solvent is what does the dissolving. The other part is called the solute. The solute, there's less of it. And this could be any number of things. For the purpose of this demonstration, we're going to say it's sodium. It could be glucose, it could be potassium, it could be any number of things. But in this case, we said this is a sodium solution, and what that means is sodium dissolved in water. And when we talk about solutions, we give it a percent or some other means like molar or something by mass, something like that. So we're just going to say this is a 20% sodium solution. What that means is if we were to look at this solution, 20% of it is sodium. 20% of it is the solute. The rest of it is the solvent, typically water. So how much water is there? Well, there would be 80%. There's always going to be 100% of solution. Whatever type of solution it is, in this case 20% sodium solution, that tells you how much solute there is. The rest is always going to be the solvent. But let's say on the other side of this membrane, there is a 30% sodium solution. So that tells me the solute, which again is sodium, there is 30% of that solution is made of sodium. Which also tells me that the solvent, water, how much water is there? 70%. Okay, so we've done that. Now let's go ahead and give these solutions a name. Let's just say the one on the left is solution A, and the one on the right is solution B. Once we've done that, once we've defined our solutions, we can start applying some more technical names. We can start talking about something called tonicity. Tonicity is a way of comparing two solutions to each other. Now, what we do when we're comparing two solutions to each other is look at the solute. Over here in solution A, I have 20% solute. In solution B, I have 30% solute. So what I could say, using non-technical terms, is B has more solute than A does. But the way that we actually say this in a laboratory is using something called tonicity words. We say B is hypertonic to A. B is hypertonic to A. B by itself is nothing. We have to compare one solution to another. Specifically, we compare one solute of one solution to the solute of another solution. Whichever one is greater, we say is hypertonic to the other. Hyper means greater than, more than, above. So 30 is greater than 20. So B is hypertonic to A. Another way that we could say this is A has less solute than B. So what we would say is A is hypotonic to B. Hypo means less than, under, beneath. 20 is less than 30. The way that I tell people to keep these straight in their heads, if you think that you're going to have trouble with that, hyper, more than, excess, greater than. A hyperactive child has more active in their little body than someone else. So hyper, excess, more than, greater than. Hypo, we said, means less than, beneath, under. Hypodermic needles go under your skin. Hypo, below, less than, under. But let me erase this and give you another scenario. let's say instead of these numbers, and we'll come back to these numbers in a moment. Let's say instead what we have is both of them are 20% sodium solutions. They're equal to each other. So what we say is A and B are isotonic to each other. A and B are isotonic to each other. Iso means equal. So A and B are isotonic to each other. But let's go back now and see why all this is important in the first place. Let's reapply our numbers. So A, again, was hypotonic to B, and B was hypertonic to A. Now, why is this important? Well, we said this is a selectively permeable membrane. Now, typically when we hear that, it will say this membrane is permeable to, and it will tell you what can cross. But if it does not tell you what can cross, that means water and nothing else can cross that membrane. So here, this is a membrane that is permeable to water and nothing else. And if I put solution A on that side and solution B on the other side, and look to see what's going to happen if we let this sit. Well, remember, everything likes to go from high concentration to low concentration. So this sodium wants to move over from B to A. But it can't do that because this membrane is not permeable to sodium. Instead, water can move because it's 80% here and 70% here, and water will move from high to low. Now that is correct. A will lose water and B will gain water. But there is a different reason why that water is moving. So here I'm going to describe exactly what's happening at the molecular level, and then next semester in Bio 139, when we talk about solutions again, I'm going to know who had me for Bio 137 because of a phrase that I use that everyone remembers. Okay, solutes pull water towards themselves. That's just something that happens. They're like little magnets for water. So what I say is solutes suck because they suck water towards themselves. And they suck all water towards themselves. So this sodium over here in B is pulling this water towards itself. And this solute over here is pulling this water towards itself. But it's not just pulling over here on the left side. This 20 is not just pulling on that water in A. It's also pulling this water from B. And this solute over here on side B is also pulling water from side A. So water is getting pulled from B to A, and water is getting pulled from A to B because those solutes are sucking the water towards themselves. But if we go back and we look at those solutes, where is the greater solute concentration? Which side has more solute? So which side is sucking harder? Well, B has more solute. So more water will move from A to B. Now there is still some water moving from B to A, but not much. So if we were to let this sit and we came back an hour later, A would actually lose volume and B would gain volume. And how long would it keep doing that? Well, what actually happens is water gets pulled from A to B. The water concentration goes down and the solute concentration goes up. Over on side B, as water enters, the water concentration goes up and the solute concentration goes down until eventually they have the same concentration on each side. So osmosis occurs, and the result is the hypotonic and the hypertonic solutions are no longer there. We have two solutions that become isotonic to each other. Let that sink in. The osmosis occurs and it causes the hypotonic and the hypertonic solution to change until they are isotonic to each other. Now, once they are isotonic, let me erase this. Once they are isotonic to each other, I'm just going to write iso, not even worry about the numbers. Once they are isotonic to each other, what happens? Well, if we were to just watch it, it would look like that water level doesn't change anymore. But does that mean nothing is moving? No. It's kind of a trick question that shows up on exams now and then. When two isotonic solutions are separated by a selectively permeable membrane, water is moving in both directions. But it's moving in both directions at an equal rate. We have reached equilibrium. Now the water levels stay just where they were, but molecules of water are moving from A to B and from B to A, but at the same rate. Now, at first it may seem like this isn't super important for an anatomy class, but I want to show you the other way that this can be presented. Let's say I have a cell that is down in a beaker of water. Now, if I have this cell down in a beaker of water, let's say I put some particles in this water. Let's say I put some solute in this water. So instead of just saying I have a cell in some water, now I say a cell is placed into a hypertonic solution. A cell is placed into a hypertonic solution. Well, remember, a solution by itself cannot be hypertonic or hypotonic or isotonic. It's always two solutions compared to each other. So where is the other solution here? It's inside the cell. So if the solution is hypertonic to the cell, that means the cell is hypotonic to the solution. If I put a cell down into a hypertonic solution, what will happen over time? Where are there more solutes? Well, that's what hyper means. So there are more solutes in the solution, and what do solutes do? Solutes suck. They're going to suck water out of this cell, and the cell is going to shrivel up. It's going to do what we call crenate. And if you look in your lab manual, that's what we see right here. The cell has crenated. It has lost liquid. And that is a term you need to know. It crenates. But what happens if a cell is placed into a hypotonic solution? That means the cell is hypertonic. So it has more solutes inside, and those solutes are going to suck, and they're going to suck water into the cell. The cell is going to swell up like a balloon. And if it swells up enough, just like a water balloon that's overfilled, what's going to happen? It's going to burst. We say it's going to lyse. Lyse means to rip apart or to break. So, this kind of thing can be really important if you are ever going to be giving an IV to somebody. In an isotonic solution, we can see here these arrows show that liquid is moving in and out of the cell at the same rate. In a hypertonic solution, liquid water is getting pulled out of the cell and it crenates. In a hypotonic solution, water is moving into the cell, so the cell will start to swell. If it's too much, then not only will the cell swell, it will lyse, it will pop. Now, the way that I always describe this, just to kind of drive everything home in my in-person lab, is I will say, this is important for if you are ever lost at sea and you end up on a desert island. So we're surrounded by seawater. And what are you supposed to never do if you're lost at sea or you're lost on a desert island? You don't drink the seawater. No matter how thirsty you are, you don't drink the seawater. Why not? Because seawater is hypertonic to our body tissues. And when that seawater passes through our stomach and gets to our small intestines, it's hypertonic to the point that it pulls water out of our small intestines with such force that it actually rips apart a lot of those cells. And when you die from seawater, it's a not pleasant way to go. What actually happens is you die of bloody diarrhea because your intestines have been ripped open. So that's why you don't drink seawater. Now, in person, I would have set up this kind of thing to demonstrate osmosis. Actually, it's on a separate sheet. So we have this sheet. Now, most of this you can fill out on your own. I'll talk about it in just a moment. But I would have set this thing up in lab. So I'm going to go ahead and tell you what to put in each of these boxes, and I'll describe what I did. So what I do is I take a beaker and I fill it either with distilled water or deionized water, which is just pure water with nothing in it at all. That would be a 0% sucrose solution. Or I take water with sucrose dissolved in it, and we dissolve it at a rate of 40% sucrose. So some of that solution is put down into something called a dialysis bag. A dialysis bag really is just an artificial cell. And then I seal up that dialysis bag and I put it down in a beaker. So what we end up with is this setup. A beaker with a solution and a cell down inside of it. And we have all three scenarios. And then we watch to see what happens. So in the first setup, we have a 40% sucrose solution in a bag. So in this first box, you would write 40% sucrose solution in that bag. And we put that fake cell down inside of a beaker that is pure water. So it is a 0% sucrose solution. 40 in the dialysis bag, 0 in the beaker. Now, what we would say is the bag, that cell, is blank to the beaker. What would we say? The bag is hypertonic to the beaker because the bag has 40. The beaker has 0. 40 is greater than 0. The bag is hypertonic to the beaker. Now, I've got some notes here from when I did this back in the spring. And at the start, I weighed that bag before I put it down in the solution. And the bag was 26 grams. So go ahead and put that here. 26 grams. And then I let it sit for almost the entire class. At the end of class, I took the bag out and I weighed it again. What do you think happened as that bag sat down in a solution and the solution had less solute. What do we think happened? Well, at the end, I weighed that bag again, and it was 30 grams. So at the start, it was 26. At the end, it was 30. So what happened? It gained 4. Why did it gain 4? Because the solute in the bag was higher than the solute in the beaker. The bag sucked water in. The solutes in the bag sucked, pulling water into the bag. Next, I had a bag with pure water inside of it, so 0% sucrose in a bag. And I put that bag down in a beaker where there was 40% sucrose. 0 in the bag, 40 in the beaker. The bag was what to the beaker? The bag was hypotonic to the beaker. And I weighed that bag at the start, and it was 24 grams. At the end of class, I weighed it again. What do you think happened? At the end, the bag was 21 grams. It lost 3 grams during the class because the solutes out here in the beaker sucked water out of the bag. And the last scenario was I had pure water in a bag, 0%, and pure water in a beaker, 0%. So 0 in the bag, 0 in the beaker. The bag was what to the beaker? The bag was isotonic to the beaker. Now, at the start, I weighed it, and it was 27 grams. The bag was 27 grams. At the end, when I weighed it, what do you think that it was? It was also 27 grams. This one is a little bit tricky. Usually when we do this one, sometimes it's a little bit more at the end, but that's just because when I put it down in the beaker and I take it out to weigh it, there's water stuck to the outside of the bag, just where it was sitting down in the water. So that water makes it weigh a little bit more. In the spring, it worked out really well. It was 27 at the start and 27 at the end. So there was no change in weight. Does that mean nothing moved? No, it doesn't mean that at all. Water was constantly moving in that bag and out of the bag. It was just doing it at the same rate. So the bag did not gain or lose water. Now you should be able to answer these questions on this sheet based on everything talked about so far. I do want to point out something that I've got highlighted here in this image. For number 5, it looks like it's looking for one answer, and I think it was a typo. So the way that it's written, there should be 2 answers here. One says, which of these items would move the slowest? And it also asks which would go the farthest. Well, the one that moves the slowest will not go very far at all. The one that moves the fastest will go the farthest. So you'll have 2 answers here the way that this is written. And again, that's just based on the things that affect the rate of diffusion. Moving on, the next type is filtration. Filtration is passive transport, but it's both water and solutes that are moving across a membrane. But here it's something different that's causing them to move. Here it's hydrostatic pressure that's causing them to move. So it's pressure of the liquid, of the solution against a membrane, and it is just pushing its way through. So it's moving from higher hydrostatic pressure to lower hydrostatic pressure. And the water and the solutes that are dissolved in it both cross the membrane. Now it gives you an example here of coffee moving by filtration through the pores of a coffee filter. And that's true, but there is a much better example of filtration that actually occurs in the body that's going to be really important next semester in Bio 139. Your kidneys use filtration to cleanse your blood. In your kidneys, in each kidney, there are 1 million little things called a glomerulus. There's about 1 million in each kidney. It's a very specialized blood vessel, and as the blood moves through, it is filtered. Filtration occurs, and liquid with dissolved substances are filtered out of your blood. So just know filtration, it's passive, and it's hydrostatic pressure that's causing it to occur.